10 days. Double digits carry psychological weight, but they also carry clinical weight. At day 10, you've crossed a threshold in several repair processes that have specific 10-day timelines in the cessation literature. Heart rate variability, a marker of cardiac autonomic function, shows measurable improvement around this stage. Arterial stiffness is decreasing. Gingival blood circulation has approached non-user levels, and something that most quit resources don't mention at all. DNA methylation patterns altered by years of smoking are beginning to show early signs of reversal at specific gene sites. That's epigenetic repair. Your body is not just clearing the chemical effects of nicotine, it is beginning to undo damage at the level of gene regulation. Day 10 is also where hippocampal neurogenesis resumes. New neuron production in the part of your brain most critical for memory formation and mood regulation. It's where circulating stem cells increase in number. It's where sleep architecture largely normalizes, hours 217 through 240. At hour 217, type Y pneumocytes, the thin flat cells that form most of the alveolar surface and are critical for gas diffusion, are being replaced through differentiation of type 2 pneumocytes. This cellular turnover restores the integrity of the alveolar capillary membrane, the ultra-thin barrier across which oxygen enters your blood and carbon dioxide exits. When this membrane is restored to proper architecture, gas diffusion efficiency improves. Not dramatically, but measurably. At hour 218, heart rate variability is increasing. HRV is the variation in time between successive heartbeats. High HRV reflects a healthy, responsive autonomic nervous system. The parasympathetic and sympathetic branches in dynamic balance. Chronic smoking suppressed vagal tone and compressed HRV. Lower HRV is associated with increased all-cause cardiovascular mortality in population studies. At day 10, vagal tone is recovering. The heart is becoming more responsive to moment-to-moment demands rather than running at a chronically elevated sympathetic tone. At hour 219, pulmonary dendritic cells, the antigen-presenting cells that bridge innate and adaptive immune responses in the lung, are recovering their function. Improved dendritic cell activity enhances the specificity of the adaptive immune response to respiratory pathogens. Your lungs are becoming immunologically sharper. At hour 220, pulse wave velocity, a clinical measure of arterial stiffness elevated in smokers, is decreasing. This is a direct consequence of improved endothelial function and recovering vascular compliance. Decreased arterial stiffness reduces cardiac afterload. The heart has to push against less resistance with each beat. Blood pressure may be showing measurable improvement by this point. At hour 221, the one that most people don't expect, epigenetic repair is beginning. Smoking alters DNA methylation patterns at specific CPG sites across the genome. Methylation changes at gene promoter regions can silence tumor suppressor genes, alter inflammatory signaling gene expression, and dysregulate multiple biological pathways. These are not the same as DNA mutation. They're reversible changes to gene regulation. And at day 10, research shows early signs of reversal at specific methylation sites. The gene promoter regions associated with tumor suppression are beginning the slow process of epigenetic normalization. This process will continue for months and years. But it is measurably beginning now. At hour 222, gum tissue color is transitioning toward healthy pink coloration. Restored microvascular perfusion in the gingiva is approaching non-smoker levels. At hour 223, basal metabolic rate is adjusting downward by approximately 4 to 8%. Nicotine's thermogenic effect has fully cleared. This is physiologically normal and healthy, though it may contribute to modest weight changes. The metabolic rate your body settles into now reflects your actual baseline. At hour 224, the biochemical composition of airway mucus is normalizing. The ratio of gel-forming mucins, MUC5AC and MUC5B, is returning to appropriate proportions. Normalized mucus viscoelasticity makes it more effective at trapping and transporting inhaled particles without becoming too thick to clear. At hour 225, prefrontal executive function continues to recover. Working memory, cognitive flexibility, and inhibitory control all depend on prefrontal dopaminergic and glutamatergic signaling. These systems were disrupted during acute withdrawal. At day 10, they are measurably more functional than they were at day 3. At hour 226, hematopoietic and mesenchymal stem cell mobilization is increasing. Without nicotine-mediated bone marrow suppression, stem cell release into circulation has increased. These cells are being deployed to tissue repair sites across the body simultaneously. The body's regenerative machinery is running at fuller capacity. At hour 227, the cyclooxygenase pathway is normalizing. This matters because it shifts the balance of prostaglandin production away from pro-inflammatory prostaglandin E2 and towards cytoprotective prostacyclin. The body is tilting its chemistry toward tissue protection rather than inflammatory response. At hour 228, laryngeal mucosa and vocal fold epithelium are recovering. Edema and inflammation of the vocal folds is reducing. Voice quality is improving. Hoarseness diminishes. Vocal range increases. This is a change that others around you may notice before you do. At hour 229, the complement cascade, a key arm of innate immunity that was chronically activated by smoking-induced tissue damage, is returning to baseline activity. Normalized complement function means the immune system is no longer in a state of constant inappropriate activation. It responds to real threats rather than chronic irritant noise. At hour 230, capillary density in peripheral tissues is beginning to increase through angiogenesis. New capillaries are forming in response to improved VEGF signaling, vascular endothelial growth factor, which smoking had dysregulated. More capillaries mean improved nutrient delivery and waste removal at the tissue level. Physical stamina during activity is noticeably improved at this stage. At hour 231, noradrenergic neurotransmission from the locus coeruleus is normalizing. The hypervigilance, the exaggerated startle response, the general nervousness of early withdrawal, those were partly driven by locus coeruleus dysregulation without nicotine's modulation. At day 10, norepinephrine balance is restoring. Attention regulation improves. The nervous system settles. At hour 232, gastric mucosal blood flow is improving, and prostaglandin-mediated protection of the stomach lining is recovering. Peptic ulcer risk, elevated in smokers, is declining. At hour 233, plasma levels of ascorbic acid, alpha-tocopherol, and beta-carotene are rising. These antioxidants, chronically depleted by the free radical load of cigarette smoke, are being rebuilt. The body's antioxidant reserves are being restocked. At hour 234, nicotinic acetylcholine receptor downregulation is accelerating. Receptor density is declining measurably toward non-smoker levels. Fewer binding sites means fewer craving signals. The situational cravings that remain at day 10 are contextual, triggered by environmental cues, rather than the constant physiological demand of early cessation. At hour 235, peak oxygen uptake during exertion is improving as pulmonary gas exchange, oxygen carrying capacity, and peripheral oxygen extraction work together. Exercise tolerance is measurably higher than it was at day 1. At hour 236, cutaneous wound healing speed is approaching non-smoker rates. The healing machinery, tissue oxygenation, fibroblast function, collagen deposition, has normalized enough to make a measurable difference in how quickly the body repairs physical damage. At hour 237, hippocampal neurogenesis is resuming. This is one of the most significant processes occurring at day 10. The hippocampus, and the brain structure most critical for memory consolidation and mood regulation, undergoes ongoing neurogenesis, production of new neurons in the dentate gyrus. Chronic nicotine exposure and the stress of withdrawal suppress this process. At day 10, it is resuming. New neurons support memory formation and contribute to improved mood regulation over the coming weeks. At hours 238 and 239, multiple organ systems are simultaneously engaged in repair, compounding in their effect. Sleep architecture has largely normalized, appropriate proportions of NREM and REM sleep efficiency approaching 85%. The sleep you're getting now is qualitatively different from the fragmented REM-suppressed sleep of your smoking period. At hour 225, the clinical record notes something important for understanding day 10. Executive function mediated by the dorsolateral prefrontal cortex is continuing to recover. Working memory, cognitive flexibility, and inhibitory control are measurably stronger than at day 3. This has a specific practical implication. The prefrontal cortex is the seat of impulse control, the part of the brain that evaluates competing options and overrides immediate desire in favor of longer-term goals. Nicotine withdrawal temporarily degrades this function. During the first 5 days, your ability to resist urges was physiologically compromised. The cravings you were fighting were neurologically more powerful than usual, and your resistance circuitry was simultaneously impaired. At day 10, that asymmetry has resolved. Your impulse control is functioning normally. The remaining cravings, which are infrequent and situational, are meeting a fully operational prefrontal response. When the trigger fires, you have the full processing capacity to route the response differently. This is not metaphor about willpower. It is the functional restoration of dopaminergic and glutamatergic signaling in the prefrontal cortex, measured in clinical research. Hour 240, 10 full days without nicotine. Gingival blood circulation has approached non-user levels. Nicotinic acetylcholine receptor downregulation is accelerating. Pulmonary function shows measurable FEV1 improvement. Systemic inflammation has substantially resolved. Multiple organ systems in simultaneous active repair. The double-digit milestone reduces relapse probability substantially, not because of willpower or psychology, but because the pharmacological foundation for craving has been progressively eroded. Receptor density is declining. The GABA system is balanced. Dopamine baseline is normalizing. Sleep is restored. The body is no longer in chemical distress. The sprint is not over. 4 days remain. But from day 10, the trajectory is firmly established. Day 11 is the identity phase. Nicotinic receptor density approaches non-smoker baseline. Platelet aggregation is fully normalized. Synaptic pruning of the old smoking habit loops is actively underway. The brain is beginning to structure